So thanks very much. I've actually had a great day today and had some really nice talks with people. And before I get started on telling you what I've been doing for the last 10 years, I'd just like to acknowledge the support of NIH. And actually, this research has been done pretty much independently of my graduate students. My graduate students go off and do their own projects on different species. And but I have had an army of undergraduates that have helped me do a lot of the field work, and a couple of postdocs as well. So what I'd like to do is I'd like to start with a very basic observation, that when we talk about phenotypic variation in populations, most of us think of, okay, there's an influence of genetics and there's an influence of environment, and in fact those two things interact. But what I'd like to do today is to broaden that discussion, and I'd like to add two more pieces to that. One piece I'd like to add is age and its influence on phenotype, and then the other part of it is cohort. And so let me just sort of go into depth a little bit more on those two pieces. So first of all, age. So when I got started on this work in looking at the age and the influences on life history traits, it turns out that this was about 15 years ago, and there wasn't much information from natural populations. And if we want to define aging as an increased risk of dying as you get older or a decrease in physiological performance as you get older, we knew that in controlled environments, from humans to Drosophila, nematodes, and mice, we find those types of patterns. We find that as individuals get older, they decline physiologically and they increase in their mortality. But we didn't have much information at that time from natural populations. In fact, most people had dismissed natural populations and said there's no such thing as aging. Well, in the intervening 15 years or so, we do have some good information. And I've just pictured a couple of these experimental systems where, in fact, we are now getting much more information. And I can go from David Resnick's guppies to the swan, which actually shows a decline in reproduction as individuals get older. And then there's the sheep and the roe deer. And in all of those cases, we do have some information that, in fact, aging is something that's important. It does influence phenotypes in natural populations. But I've got a little green thing in the corner there. It says, what about plants? Okay, and in fact, before I started this work, we really didn't have any good information. Plants had always been looked at as these things that just live for too long a period of time, and we'll never be able to understand, the influence of age on phenotype. And so that's what motivated me to do the design of the experiment that I'm going to tell you about today. But before I get to that, I also want to throw in this other component that I think influences phenotype, and that is cohort effects. So cohort, basically, a group of individuals born at a particular time at the same time. Now, we all know that environment influences early development, which then influences survival and reproduction, and ultimately an individual's fitness. Well, cohort effects is this idea that individuals who are born at the same time may share a particular environment, which actually may influence their phenotype differently than a group of individuals born at another time. So, for example, low nutrition. Okay, low nutrition may be a common factor that is influencing all individuals at a particular time. They're born a particular year. They have slow early life development, and then depending on the environment, they may have the possibility of catching up, maybe not. But very often these low nutritional influences of the environment actually carry through and influence survival and reproduction at the latest ages. Okay, so this is what I'm going to talk about. We know, again, that in large mammals there's good evidence of cohort effects, and also in birds like the oystercatcher and in the common lizard there's some good evidence of cohort effects. But again, we don't know about plants, okay? And so my design of my experiments is really to get at these effects of age and the effects of cohorts. And so this is where I'm going today. is that I'm going to look at, today I'm going to tell you about a longitudinal study that looks at lifespan, mortality patterns, and reproduction patterns. And I'm going to add to this looking at the influences not only of genetics and environment, but also of age and cohort. Now, before I go on, I want to be sure that you can appreciate why it is that plants might be different, okay? And there are a couple of reasons that we might expect them to be different. One of them is that, in fact, many plants actually show indeterminate growth in the sense that they can continue to get larger as they get older. As they get larger, they also increase in reproductive output. And if you're older and you have higher reproduction, you actually may be violating a lot of the theoretical foundation about why we have aging, okay? Because in fact, it may be more valuable to be older. And so this idea of indeterminate growth may influence the patterns of aging in plants. The other thing about plants that may also influence these patterns is that they're incredibly plastic, okay? And this is you think about a leaf on a plant. Well, it turns out that we can talk about not only plant lifespan, but we can talk about leaf lifespan. And in fact, new leaves old leaves die, new leaves are born, and so there's this turnover of body parts, which might influence these age-specific patterns of mortality and reproduction. And then in plants also, because of this plasticity and these growth patterns and so forth, the question is, are the patterns age-specific or are they size-specific? And so we have to start to tease some of that out. Now, this lovely plant that you've been looking at is Plantago lanceolata, and that's the plant that I have used in this experiment, and it's beautiful for a number of reasons. It's not so beautiful in the sense that it is a weed, and so it's found all over the world. But the lovely thing about it is that we can easily define what an individual is. And actually, this is one of the reasons that plant demography has been slow to get started, is because many people have talked about the difficulty of identifying individuals. So we can identify individuals. The other nice thing about this species is it stays green all year round. So we can do demography, we can go out, and we can follow individuals. They don't go dormant in the winter and then come back in the spring, and some of them are lost. And we don't have issues about mark for capture, not only because there's no dormancy like that, but also a plant stays still. It doesn't move around. And so you go out and the plants are still there, and your individuals are still there when you go back out. A natural population is actually a field. Okay, it's a mown field. That's what Plantago likes. This idea of wind pollination is also important, because what I did was it gave me the opportunity to do crosses. It doesn't self, and so it was easy to do crosses and to do some quantitative genetics. And the other important thing when we think about a model for looking at patterns of mortality and lifespan is that we want to decouple these two things. Okay, many common species like common experimental organisms like Arabidopsis, actually, you turn on reproduction and you're turning on the processes that lead to the death of the individual. Here we've got an interopair species, and so that we don't have those problems. The mean lifespan is about 2.5 years, but as you'll see, individuals can live a lot longer than that. And we also had a lot of information before I started this study. Okay, so very briefly, what I want to do is I want to tell you about the design of the experiment. And so up here in the corner, I have a picture of the field site. And the field site here is actually remember I come from the University of Virginia. And so Thomas Jefferson is still a big figure at the University of Virginia. And this is actually the site where Thomas Jefferson's father had a home, and it's where he was born. And so this is a historic site. And so it's a great place, because I also know that there'll be no building put on this, and so I can set up a long-term field study. And the study that I'm going to tell you about has been going on now for 11 years. So I went out to this field, and we went out, first of all, and we dug up individuals that were growing in the same field, because of course I want to go out and I want to understand the role of natural selection in shaping these patterns. So I want to look at individuals that are coming from their ancestral field. So I went out, found Plantago individuals that are out there, dug them up, brought them into the greenhouse, cloned them up. In other words, I ripped them apart and made genetic copies. And then what I could do is do some crosses, okay? Designate some individuals as male, some as female. It's a hermaphrodite. and then just rub them together, put a little bag on them, and then you've got your seed to do quantitative genetics. So we planted the seeds, and so then we've got these seedlings growing up, and we grew them up into so they were about 6 weeks old. And then this is where the army of undergraduates comes in. Then we get out into the field, and we have a randomized block design, and we're putting individuals out into the field in a regular grid design. And each individual seedling is planted and with a twist tie and a identification label. And so here you have, the one individual. So this is actually one individual. We actually planted over the course of 3 years. We planted 30,000 individuals. Okay, so this is a big study, and some of these numbers that I'm going to tell you about are because we're actually partly influenced by Jim Carey's work, because it's this idea that if I want to understand age-specific patterns and get out to those later ages, I need to start with large numbers, because of course there's always this background mortality in a natural population. So you have to start with large numbers to be able to get out to these late ages and really have a good mathematical understanding of what's going on at late ages. Okay, so that's the design. Now, so here are more details about exactly when we did those plantings. So in fall of 2000, we planted the first cohort. I'll refer to that as cohort 1. And so we've followed that through now till 2011. Cohort 2 was planted in the fall of 2001. Cohort 3 was planted in the spring of 2002, and cohort 4 in the fall of 2002. So we planted 10,000 here, 10,000 here, 5,000 here, 5,000 here. And so we've got these 4 cohorts out there. Well, the most important thing here, and actually it's a take-home message from the whole seminar, is that you actually need this kind of design to understand age-specific patterns. Because if you only have one cohort and you're following it, and then mortality starts to go up or reproduction starts to go down, well, you can't make the conclusion that in fact there's aging in this population, because it might have been, global warming is happening, okay? And so that's why you've got, the increase in mortality. And so it's only with this multiple cohort design that we can actually tease apart the environment and age. And so on the one hand, I can follow individuals all the way from birth here all the way until they all die. And I can follow a cohort longitudinally. But I can also follow individuals cross-sectionally. So at any one point in time, I can go out into the population and find 4 different age groups. And so I can say, under the particular environmental conditions today, how are these individuals of different ages doing? And so that's the way that you can sort of tease apart whether or not the changes that you're seeing longitudinally are actually due to environmental changes or, in fact, are due to age-specific changes. So when you throw out individuals that died in their first few months and so forth, I have complete information for about 23,000 individuals. Now, as of about a month ago, out of these individuals that were planted, I only have about 150 left. Okay, so most individuals have died. And so maybe 5 years from now, I'll be able to talk about the oldest old Plantago ever on Earth, and I can, have an interesting story about them. But for now, what I want to tell you about are these longitudinal patterns. There's one other part of the design that's very important here, and that is I told you I did crosses, and the genetic structure of each of these cohorts is exactly the same. Okay, so if I find that there are differences in cohorts, it's not because there are differences in genetics. They all started with the same genetic structure, which gives me lots of fun power with respect to looking at the effects of genetics and so forth over time. Okay, so the traits that I'm going to talk about today are mortality. We go out every 30 days and look at whether or not individuals have died. Size, today I'm going to talk about looking at size every 6 months. And then reproduction, which is a yearly trait. And so we have up to 11 years of that. Okay, so what's happened? First of all, this is a basic survivorship curve where I've got age on the x-axis and survival, the percent survival. And by the way, please note when I'm showing you the results here, sometimes, because I said I like to look both longitudinally and cross-sectionally, sometimes I'm going to have age on the x-axis and sometimes I'm going to have date, okay? And so it's important to just be aware what I'm looking at, what you're looking at here. So with respect to survival, what you can see is that, well, there's something funny going on, sort of parallel patterns, but sort of a delay in many senses. So here's cohort 1, the blue one, and right around 40 months or so, when there was about 80% of the population was alive, and then a lot of them started to die. And you can see that the same thing seems to be occurring in the other cohorts, but at earlier ages. Okay, so I actually like to look at it with respect to not only I like to look at it with respect to mortality instead of age. Okay, so I'm going to flip everything around, and I'm going to look at this with respect to mortality by date. And what you find, this is exactly the same data, but just plotted in a different way. And so what you see here is that cohort 1 is going in, cohort 2 is the red, and cohort 3 and 4 are the pink and the green. And that decline, remember here now we've got date, okay? That decline all happened at exactly the same time, okay? The environment changed. All individuals, and no matter what their age, they all responded with high mortality. And I consider this as a reflection, as an ecologist, I consider this as a reflection of stress. Something in the environment occurred causing a stressful situation. Lots of individuals died. And in fact, mortality stayed up at a pretty high rate. This is mortality rate for every 6 months, by the way. Mortality stayed up pretty high for a long time. And this first analysis, I'm just going to, in a minute, I'll extend this, this graph, but this first analysis was done at the end of 2007. Okay, so what do I have here in these? There's got some asterisks up there. Well, this asterisk, this first asterisk here, is pointing out the fact that cohort 2 had a higher mortality, significantly higher mortality than cohort 1. This was the first time that cohort 2 had ever undergone reproduction, so they had a higher mortality. But what I'd really like to focus on are up here, where we have high mortality in the population, and we actually also have a difference between the older cohorts and the younger cohorts. And each place I have an asterisk there suggests that either cohort 1 or cohort 1 and 2 had a higher mortality than the younger cohorts. And this is what I would like to call condition-dependent aging. Okay, when mortality is low, or at other times, you can't really tell the difference between older individuals and younger individuals. But during these stressful conditions, when mortality is high, the older individuals are doing worse. Okay, so it suggests that there must be some sort of physiological reason that individuals that are older are not being able to cope with this stressful environment. And this was kind of neat because this was the first indication that you can actually separate individuals out. This is completely different types of mortality curves than anyone has found because, of course, mortality is so influenced by weather conditions in the natural population. So what I wanted to do, first of all, was to, if I can go back a second here, I was really curious to say, let's look between 2003 here and 2006, when mortality is really high, could I identify any ecological factors that would explain this high mortality? Well, I looked at maximum temperature, minimum temperature. I've plotted every year here by month, and there were no differences across those years. The only thing I could find was that if you look at cumulated precipitation over the year, it turns out that this solid red line is 2003. That's exactly when mortality started to go up it really steeply. And in fact, what happened in 2003 was that we had so much rainfall that, in fact, plants love rainfall. So every other plant loves rainfall. Okay, and so what happened was the whole community shifted. There was a big change in the community, and so other individuals, other members of the community grew up, and so basically the idea here is that high rainfall can induce changes in the community. Well, Plantago, I told you, grows in fields, mown fields, and that sort of thing. And so here is an experimental individual down underneath all of this. And Plantago just wasn't happy. And in fact, I had a student who looked across the field at different blocks to understand, what were the differences between the blocks where there was higher mortality and low mortality. And there were two things. One was the amount of light that was down at the level where Plantago was, low light, low survivorship. And the other thing was waterlogged soils, and Plantago didn't like that as well either. So there was this shift over time. Of course, then we got back to the regular weather patterns and so forth, and over a 3-year time, mortality then went down. This is an update of these mortality curves. And so mortality was relatively low, but this was the only period right here where there were differences in cohort performance with respect to mortality. And what's happened over time then is that the cohorts have been very synchronous. There was a peak here at the end of 2010, and that peak was caused by just the opposite. In fact, it was caused by low rainfall. Okay, and under low rainfall conditions, none of the plants, no matter what their age, did very well. And so there was quite a spike here in the mortality rates. Okay, so we can talk about mortality rates, but we can also talk about lifespan. Okay, and I have from doing an analysis of lifespan in this population, I've found that there are significant influences of block, in other words, where you are located in the field, significant influences of cohorts. And so here, what you can see is, here's cohort 1. Cohort 1 had the longest lifespan, a median lifespan of 39 months, a median lifespan in cohort 4 of 20 months. So that's 19 months difference, really big. But that makes sense, because remember, this spike in mortality was, cohort 4 was much younger, okay? So it just makes a lot of intuitive sense. But what it did was it clearly influenced the numbers of individuals that lived to the later ages. Found an influence of sire. And one of the interesting things was that, taking me back to some work I've done earlier on maternal effects, it turns out that, in fact, there was also a greater influence of dam. In other words, there was some influence of maternal effects on lifespan here. And some of these other interactions I just want to show you here. So I have, first of all, the influence of block that I've just mentioned. So block 2, for example, all 4 cohorts here that are plotted, all 4 cohorts do well, and for example, in block 2, whereas in block 9, there's a major difference in performance, and all 4 cohorts do much worse. Now, there was also an interaction between block and cohort. In other words, it's not only that there's an influence of what year you're born, but also where in the field in a particular year that you're born. So in other words, different parts of the field were advantageous for different cohorts. So for example, in block number 4 here, we have cohort 1 doing better than 2, then 3, then 4. But in block 12, in fact, cohort 4 did relatively well. In fact, cohort 4 did better than cohorts 2 and 3. So in other words, these spatial dynamics that are going on actually are varying across years of establishment. And then finally, I show here that that there are differences in dam. For example, mom number 7, cohort 4 did particularly well. You can see, by the way, that cohort 1 is always doing better, but cohort 4 did nearly as well for mom 7. But mom 21 actually not so well. Okay, so there are these dam effects that are influenced by different cohorts. Okay, so what I also wanted to do was I wanted to say, who are the individuals who are living to the really late ages? Okay, and one of the nice things, and again, this is sort of, I sort of feel like this is one of the cool threads of this experiment, is that because I have complete lifetime data for so many individuals, I can do a retrospective analysis, and I can say, okay, who was successful later on, and how did they differ from those who weren't successful, and what was their early life performance like. And so I did I've done a logistic regression looking at early life reproduction, early life size, and early life growth. And what I found is that your performance right here during that initiation of the stress period was really important, determining whether or not you were going to be one of those individuals that survived out to be the oldest the oldest 20%. And what I found was that, in fact, it was growth rate. So your growth rate during that initial period of stress determined was an important determinant of whether or not you're going to live to be the oldest old. So as you can see here, first of all, so this dotted red line is zero growth. Okay, so anything over here is negative growth. In other words, you're shrinking, and anything over here is you're growing. Okay, and so clearly it was advantageous to be growing. Okay. And this was over here. We have the probability of surviving to the oldest ages for any given growth rate. And so clearly there's a difference here. What I've done is I've plotted the two, the oldest and the youngest cohort. And what I'd like you to see is that the youngest cohort, cohort 4, they're nearly all on this side of that red line. Okay, most of cohort 4 was growing, whereas a large proportion of cohort 1 was actually shrinking during this time. And if you look at the growth rates across cohorts, it turns out that cohort 1 had on average the lowest growth rate, and then cohort 2, but still shrinking, and cohort 3 and 4 were actually growing. Okay, so then if you go back to the mortality patterns, rats. Okay. So I don't have the mortality patterns, but remember what I said was that when we had that peak in stress and then we had the oldest individuals doing worse, well, some of it is right there in those growth patterns, in those differences in growth patterns. So what do we have here? We've got, so at this point in time, we've looked at lifespan and we've looked at mortality. We've got evidence for genetic effects, also maternal effects. We've got evidence that, in fact, some of those genetic effects interact with the environment. But we also have, clearly, we have cohort influences. Okay, it does matter when you were born. And, in fact, there are interactions between cohort and spatial environment. And then, with respect to mortality, we have this condition-dependent mortality. When mortality is high, the oldest individuals do worse. Okay, so now what I'd like to do is I'd like to shift and talk not just about lifespan and mortality, but to talk about reproduction. And first of all, I have to tell you that there is life without reproduction in this species. And it's kind of a strange thing because normally when we talk about individuals living either post-reproductively, we think about animals that have parental care. Well, of course, this species doesn't have any parental care, but they sure have a whole lot of post-reproductive life. It turns out that after an individual reproduces, they live on average about 14.7 months. Okay, that's a long time. In other words, they would have had another chance to reproduce. And then there are a huge number of plants. So about 10% of the plants, this is just cohort 1, by the way. So 10% of the plants never flowered at all. By the way, I have already taken out of this any individual who didn't even survive to the age of reproductive maturity for that first year. So I've thrown all those out. And still 10% don't reproduce at all. And they have a lifespan of about 28 months, okay? So more than 2 years. And there are some individuals who have lived 11 years and they have never reproduced. So there is, there's a non-reproductive part of life even in this species.
Are they the littlest ones?
No, they're not actually, which is kind of funny. Yeah, not expected. Okay, so to quantify reproduction here, these are the inflorescences. If you don't know Plantago, I'm sure you recognize it after looking at this particular picture here. So we can either talk about number of inflorescences or the number of seeds. But what I'm referring to as an inflorescence is this right here. Okay, so first of all, okay, so as I already told you that there's some individuals that don't reproduce at all. And so what we did was we looked to see how many individuals, how many times do individuals reproduce, okay? And it turns out that 47% of the individuals that do reproduce only reproduce once, okay? So I started this whole thing out saying, you know, I have this interopair species here. Well, you know, it's only partially so. And the other neat thing, getting onto the whole theme of looking at cohorts, is that there's variation across cohorts. And so cohort 1, you know, so this is by cohort here is the proportion of reproductive plants that are reproducing once, twice, 3 times, or more than that. And what I'd like you to see is that for cohort 1, there's a large number of them actually reproduce twice. But for cohorts 2, 3, and 4, the highest proportion of them only reproduce once. And then if you ask the question then, okay, if they only reproduce once, when are they going to do it? There's also cohort variation here. And so if you're only going to reproduce once and you're in cohort 1, about the same number will do it either in the first year or the second year. If you're in cohort 2, you'll do it in the second year. Or if you're in cohorts 3 and 4, you'll do it in your first year. So again, there's this really an important component of cohort variation here that's influencing these reproductive patterns. So how about total lifetime reproduction? It turns out cohort 1 has the highest lifetime reproduction of all the cohorts, but there is not only cohort differences, there are also spatial block influences on that. There are also sire and dam influences, no evidence of maternal effects here. And there are also, there are also some interactions. Now, with respect to the large numbers of individuals that are reproducing in huge number of individuals, huge number of lifetime reproduction in cohort 1, one of the reasons for that is that cohort 1 individuals actually were the largest early life, okay? Remember, these individuals had a longer period of time to live before that stress occurred, okay? And so it turns out that these individuals actually, this is size here, number of leaves, and this is over time. And what you can see is that cohort 1 individuals were much larger before that mortality spike occurred, and that's why they have so much higher lifetime reproduction. Now, and then we have, with respect to reproduction, there were these different. There was a block by cohort interaction, and so some blocks. So here, this block, for example, cohort number 3. Oops, yeah, cohort number 3 here actually did much better than cohorts 2 and 4, whereas cohort 3 did particularly poorly in the third spatial block, which is actually right next to the cohort number 2. Spatial block number 2. So clearly there was a block by cohort interaction, which had a major influence on these patterns. Now, you know, I like to think not only about total lifetime reproduction, but I also am interested, because of the age component, I'm interested in age-specific reproduction. And here what I've done is I've plotted for each cohort, I've plotted age-specific reproduction. This, again, I'm going to continue the curve in a second here, the curves in a second, but by date, okay? And unlike the mortality patterns, there's no particular clear pattern with respect to date for the age-specific reproduction. In other words, how many offspring do you produce at a particular year? But if you look at this with respect to age, the pattern is different. And so here, this is something that really is very interesting, because all the cohorts peaked in reproduction in their second year, and then they went down. Now, you know, maybe there's something to do with size, or maybe they're all peaking in size, and so there's something to do with size here. So I looked to see whether or not there was an influence of a change in size with age with respect to that paralleled these changes in reproduction. So cohort 1 here up at the top, and what I do, you've got size in the black line and reproduction in the blue line. And what I'd like you to see is that, in fact, yeah, okay, so size is increasing and then declining after age 2. And so, you know, there's somewhat of a similar pattern here in the in reproduction, but not in cohort 4. Okay. The nice thing is I've got all these replicates of different cohorts, and in fact, in cohort 4, which had that same pattern, in fact, size in the black here is continuing to increase even though reproduction was going down. Okay. So I do think that this pattern of a peak in reproduction is actually suggests that in fact there is some component of age-dependent reproduction in this particular species. The other cool thing, when we go back to connecting reproduction with mortality, is that in fact this peak of reproduction in age 2 was despite what was happening in the environment. Okay, because so here's this pattern of a peak of reproduction at age 2. And so I have reproduction by age. But then now let's go back and look at the mortality rate by date. And what I'd like you to see, and what I've done here is that with each of these arrows, I've marked where year 2 was for each of these different cohorts. So year 2 is right here for cohort 1, and here for cohort 2. And I'm sorry, this is age of first reproduction. But what I'd like you to see here is that some of these cohorts are they've got this same pattern, despite the fact that some of them are just starting to reproduce during this time of crisis, during this time of stress. And so you get these age-specific patterns despite what was going on in the environment. And this is very different from, the mortality patterns and the reproductive patterns are really different, and it's actually pretty exciting. So what's happened over time is that mortality peaked. It stayed very low for a long period of time. It did increase again in 2009 and go down, but all the cohorts are doing exactly the same thing. And so these more recent patterns of reproduction are much more like the mortality in the sense that it's much more responsive to the environment, as opposed to the early performance, which was more reflective of age. Okay, so one of the troubles with looking at longitudinally at populations, whether or not we're talking about humans or plants or anything else, is that you can actually what happens over time is that you're changing the composition of the population over time, and so that any age-dependent patterns that you see may be due to changes in the composition of the population. And so maybe the whole idea here is that maybe by the end what you're looking at is this really hardy group of individuals who can survive out there, and that might be influencing these age-dependent patterns that you can see. So I have, as I've mentioned before, I have this ideal situation where I can do a retrospective analysis, and I can go back and I can look, how did those individuals do when they were younger versus those who actually didn't survive to the later ages. And so what I'm going to do is I'm going to show you this with respect to 2 different cohorts. And first of all, the take-home message here is that in fact those individuals who died early on in life versus those who are the oldest old and the oldest old survivors all seem to have the same patterns of reproduction early on in life. So here we have the oldest survivors that are in the dotted line here, and this is for cohort 1 and cohort 4. And what you can see is that both the oldest survivors have this peak at age 2 and then a decline, and these short-lived individuals also have a peak and then a decline. And also this is also true with respect to cohort 4. The patterns are slightly different, but again, there's the patterns that are similar across the oldest survivors and then those individuals who actually died at early ages. And so we don't have any influence of changes in the composition of the population when we're looking at these particular patterns. So I also looked at this not only with respect to reproduction, but I also looked at it with respect to size. And in cohort 1, what I found was that those individuals, okay, so here we have size on the y-axis and age, and this is size as number of leaves. And what I found was that, in fact, the survivors, actually after age 2, the survivors were the largest plants, okay? But again, these trajectories with age are the same for the survivors and those who have shorter lifespans. The trajectories are the same, but they're just at different levels. If you're going to survive, you're actually at a larger size, even at early ages. Now in cohort 4, this is kind of interesting because again, there's a similar parallel, in fact, trajectory of those individuals who are going to be the survivors, who are going to survive out to be the oldest 20%, versus those who have a shorter lifespan. There's parallel trajectories here, but they're different across cohorts. Okay, and so here, from age 1 to age 4, cohort 4 is constantly increasing in size, whereas cohort 1 increased in size at age 2, but then declined thereafter. So within a cohort, though, the trajectories of the survivors and the ones who are shorter lived are the same. So, it's also interesting to think about these survivors and think, does it matter if you live longer? And in fact, there's a positive relationship between lifespan and lifetime reproductive output. And so these, what I have plotted here is a lifetime reproduction for individuals who survived to be the oldest 20% versus those who died, who had a lifespan that was shorter than the median lifespan for a particular cohort. And what you can see here is that, in fact, there is a positive relationship between living longer and reproducing more. And there's no particular trade-off. Okay, so what do we have here with respect to reproduction? So first of all, it turns out that what I talked about was that, in fact, most individuals reproduce only once. But when you do that one-time reproduction, depends on which cohort you're in, there's huge influences of cohort on reproduction. And in fact, there's also influences of location in the field and genetics. And then with respect to age, there's in fact a really interesting age-dependent pattern where individuals seem to peak at age 2 and then decline thereafter. So to pull this all together, I think that this experimental design that I had is actually allowed me to sort of tease apart things that are really kind of difficult to tease apart, and that is the effects of age, effects of cohort, and it's this multiple cohort design that has allowed me to do this. And I think, very often you read papers and people talking about aging in natural populations, but I actually look at it with a skeptical eye if there's only one cohort, because you don't know how much the influence of environment is influencing the patterns that are being observed. And then the cool thing, I think, also is this, with this longitudinal data, we actually have the power to then go back and do a retrospective analysis and see who are the survivors. How does early life performance influence the performance at late ages? And so in the bigger picture of life histories, clearly there's a lot of plasticity here. Plants are shrinking, they're growing, and a lot of the response to the environment is because of this plasticity. But we do have aging effects. We have aging effects with respect to the mortality patterns during times of stress, and we also have it with respect to reproduction. Clearly there are large cohort effects, and the oldest survivors are a particularly unique group that I think there's still a lot more to be looked at with respect to the data that I have on them. So I'll leave you with this picture here, which is basically the outline of my talk, and suggest to you that I think I've got nice evidence here of the influences of environment, genetics, age, and cohort on natural plant population. Thank you very much.
Well, that was great. You know, just a technical point here to graduate students. I've been interested in best practices in PowerPoint. This is how you do it. Really, really clean slides and really clear. So anyway, questions for Debbie here?
Yes, Drew.
So I may have missed it, but are there any traits that identify who get those to be very, very old? Either environmental or genetic or anything else? Who are these guys?
Yeah. So it depends on what cohort. So the question is, who are these individuals that are getting to be really old? Okay. So one of the things that I had there was demonstrating that in fact the oldest individuals actually were of larger size very early on in life. Remember the trajectories, the parallel trajectories across those two cohorts. I also know that that there are unique genotypes as well, and there is a strong genetic component to that. And not only that, though, the genetics, the best genotypes for one cohort are different than the best genotypes for another cohort, which is kind of cool. And we've done some population modeling where we've actually trajected out the different cohorts and actually find that over 100 generations, not 200 generations, what we find is that one particular genotype will dominate in one cohort, and two other genotypes will dominate in another cohort. So it's definitely a genetic component to it as well. Yeah.
So if there's a genetic component, it seems like it would be maybe the first cohort you select for high plasticity, just grow like mad. And then the other one's like, oh, crappy year, the stress. There should be a stress tolerator genotype. Is this possible? What's going on? That we got that kind of selection, or is it just grow like mad and just?
Well, actually, what I haven't done and I need to do is to do a selection analysis and look to see what I do know that selection for I have done an initial analysis and shown that the intensity of selection on size does vary across years. And it's kind of messy. It varies across cohorts too. And so I need to really, you're right, I need to add that to this, and then also do it at the level of genotype.
Yeah, because you said you got the same genetic background.
Yeah, no, it's a really good thing to do. Yeah.
So the relationship, let's go along that line, the relationship between longevity and high reproductive output makes me, it's tempting to read into that there's strong selection, directional selection favoring long-lived individuals.
Yeah.
And so the question is, why aren't they 20 years old?
Yeah, I don't know yet. And I've got
to be a trade-off of some sort.
There's got to be a trade-off. But Plantago's not going to take over the world, so I know that there's got to be some trade-off. But I have looked at genetic correlations across life stages because that's something I like to do, and so far I haven't found any good trade-offs. But yeah, I'll let you know. Yeah, it's good. Yeah, Rick.
Are any of these? I'm trying to sort out in my mind to what extent cohort effects and environmental effects are correlated with
So cohort effects really are early life environmental effects, but here what I talk about environment in two different ways here, and that was spatial environment and also the rainfall type of thing. But yeah, there's clearly an environmental component to cohort effects. Yeah.
And then are any of these plants allowed to interact with one another, or are they sufficient?
No, they were planted 15 centimeters apart, and so they're not influencing each other.
So none of the cohort effects could be sort of positive or negative.
No, and there's a good sized natural population of Plantago here, which we did not disturb. The only thing we did was we put a plug. We basically made a hole and we dropped the plant right into it on a regular grid pattern. So we didn't disturb the natural population, and they're not interacting with each other. They are also interplanted, so you know, the first year we saved spaces and so forth. So no, there's no chance of that actually. And density gets the density of the experimental plants got low pretty quickly just because of background mortality, random mortality. Yeah.
I was wondering if you looked at below ground biomass allocation at all, because it seems like root systems would be the sort of kind of thing you'd want to look at for a compounding factor with age.
Yeah.
Since it can kind of like carry over the root system.
Right.
activities here.
Right. Well, there's no. So I have not looked at roots here in the field, but I have a whole other experiment with a couple thousand plants in the greenhouse, because you know one of the hypotheses out there is this idea that in fact you know you weren't going to find aging in a natural population because background mortality is so high you just can't detect anything. And so I actually have had this population growing for 18 years in the greenhouse, and we repot that, and we can see. So when we repot it, we can see that in fact there is a decrease in the root biomass as individuals get older. And in fact, what we're finding is that there's a much cleaner pattern of aging going on in the greenhouse. But since they haven't all died, they're even close to die yet. I'm not going to talk about it publicly. So, but that's why I haven't included that. But in fact, we do know that biomass is starting to shrink. Yeah, so that's a good question. Yeah.
So you showed that there was a conditioned response to mortality in the first peak in 2003, but there weren't significant differences in the second peak. Do you have a reason for that? And like, could that be related to whether or not they had their reproductive peak yet? Because I think 1 and 2 had already had their reproductive peak before that first.
I think what's going on in terms of the peaks in mortality were two different abiotic things going on. Okay, so first of all, high rainfall, and then that influenced the biotic interactions going on. And so then so those biotic interactions were intense competition for a couple of years. And then the other one was the abiotic stress of low rainfall. And so one hypothesis, to sort of maybe get at what may be troubling you, one hypothesis would be that if we took an older individuals and younger individuals and we stressed them, in fact, the older individuals should do worse. And we're actually doing that experiment now. So we have, this is just in the greenhouse at this point, but what we have are older individuals, young individuals, and we're putting them under light stress, and we're putting them under water stress conditions, and we're looking at the differences there. I'd also love to be able to do another experiment where you take old individuals, young individuals, and actually put them under competitive stress as well, to sort of complete that, our understanding of what was really going on. Test that hypothesis that, in fact, it was these interactions. So that's good.
So I have a question. So, you know, the program we worked on was, they're concerned about the oldest old in humans is 85 and older. But I mean, so the extreme ages. So maybe you could comment on the growth rate as a biomarker for the ability to reach these extreme ages. Was this, maybe you can comment on that. Was that selection, or is it a cause and effect, or I mean, how does this map into ability to make it to these extreme ages?
Well, clearly the growth rate, it was the growth rate under stress, but that was actually the biomarker. And it really was this biomarker that then predicted whether or not you were able to survive to the oldest ages. And it really was, I really expected there to be, you know, some patterns of, you know, how much you reproduced in your first 2 years, may have been costly or something like that, but there wasn't any of that. It was really, it was this ability to grow during times of stress. Yeah, clearly it was a biomarker. Yeah.
Okay, one more, maybe?
I have a community ecology question. So in that wet year, these other plants grow up and start to shade these guys out, and they start to die. And that's why there's this multi-year price they pay for that single wet year. But then it disappears, or it comes back. Is that community reconverging, or is it just we've washed out the ones that are vulnerable?
I don't. It's really interesting. It basically converted. It just converted back. The community converted back to what it had been, which was this lower, scruffy type of thing instead of, you know, just everything grew up. I mean, it was really interesting even during this year where we had the high rainfall. You know, people would say, oh man, isn't it great this year? And everything's so green, you know? And, yeah, well, yeah. They goes dying. What's going on? But yeah, and so, but there's clearly, as I say, we did have nice evidence that those areas of the field that had the highest mortality, in fact, had also had the highest height surrounding vegetation and the lowest light. So yeah. Part of it is cohort, and so let me just sort of go into depth a little bit more on those two pieces. So first of all, age. So when I got started on this work in looking at the age and the influences on life history traits, it turns out that this was about 15 years ago, and there wasn't much information from natural populations. And you know, if we want to define aging as an increased risk of dying as you get older, or a decrease in physiological performance as you get older, we knew that in controlled environments, from humans to Drosophila, nematodes, and mice, we find those types of patterns. We find that as individuals get older, they decline physiologically and they increase in their mortality. But we didn't have much information at that time from natural populations. In fact, most people had dismissed natural populations and said there's no such thing as aging. Well, in the intervening 15 years or so, we do have some good information. And I've just pictured a couple of these experimental systems where, in fact, we are now getting much more information. And I can go from David Resnick's guppies to the swan, which actually shows a decline in reproduction as individuals get older. And then there's the sheep and the roe deer. And in all of those cases, we do have some information that, in fact, aging is something that's important. It does influence phenotypes in natural populations. But I've got a little green thing in the corner there. It says, what about plants? Okay, and in fact, before I started this work, we really didn't have any good information. Plants had always been looked at as these things that just live for too long a period of time, and we'll never be able to understand, the influence of age on phenotype. And so that's what motivated me to do the design of the experiment that I'm going to tell you about today. But before I get to that, I also want to throw in this other component that I think influences phenotype, and that is cohort effects. So cohort, basically, a group of individuals born at a particular time at the same time. Now, we all know that environment. So thanks very much. I've actually had a great day today and had some really nice talks with people. And before I get started on telling you what I've been doing for the last 10 years, I'd just like to acknowledge the support of NIH. And actually, this research has been done pretty much independently of my graduate students. My graduate students go off and do their own projects on different species. And but I have had an army of undergraduates that have helped me do a lot of the field work, and a couple of postdocs as well. So what I'd like to do is I'd like to start with a very basic observation, that when we talk about phenotypic variation in populations, most of us think of, okay, there's an influence of genetics and there's an influence of environment, and in fact those two things interact. But what I'd like to do today is to broaden that discussion. And I'd like to add two more pieces to that. One piece I'd like to add is age and its influence on phenotype. And then the other influence is early development, which then influences survival and reproduction and ultimately an individual's fitness. Well, cohort effects is this idea that individuals who are born at the same time may share a particular environment which actually may influence their phenotype differently than a group of individuals born at another time. So, for example, low nutrition. Okay, low nutrition may be a common factor that is influencing all individuals at a particular time. They're born a particular year. They have slow early life development, and then depending on the environment, they may have the possibility of catching up, maybe not. But very often these low nutritional influences of the environment actually carry through and influence survival and reproduction at the latest ages. Okay, so this is what I'm going to talk about. We know, again, that in large mammals there's good evidence of cohort effects, and also in birds like the oystercatcher and in the common lizard there's some good evidence of cohort effects. But again, we don't know about plants, okay? And so my design of my experiments is really to get at these effects of age and the effects of cohorts. And so this is where I'm going today, is that I'm going to look at, today I'm going to tell you about a longitudinal study that looks at lifespan, mortality patterns, and reproduction patterns. And I'm going to add to this looking at the influences not only of genetics and environment, but also of age and cohort. Now, before I go on, I want to be sure that you can appreciate why it is that plants might be different, okay? And there are a couple of reasons that we might expect them to be different. One of them is that, in fact, many plants actually show indeterminate growth in the sense that they can continue to get larger as they get older. As they get larger, they also increase in reproductive output. And if you're older and you have higher reproduction, you actually may be violating a lot of the theoretical foundation about why we have aging, okay? Because in fact, it may be more valuable to be older. And so this idea of indeterminate growth may influence the patterns of aging in plants.